Okay, so the next presentation is by Lamis Abdelhakim from PSI here on integrative phenotyping combined with modeling as a tool for predicting harvest-related traits. Where's the presentation? This one?
Yeah.
And share screen now.
Okay.
We have to share screens. Here's the sharing. Yeah.
Thank you. Hey, hello everybody, and I'd like to welcome people online. Welcome on board. Today I will be talking about a case study for the integrated phenotyping combined with modeling as a tool for predicting harvest-related trait. Can you hear me in the back? Okay, super. So before diving into the details, let me quickly recap why this project matters. As we know that climate change negatively influencing agriculture productivity, we know can influence the yield. Therefore, having integrated high-throughput phenotyping platform, enabling the breeders and us also as a scientist to screen large population and look further into different genotypes or species. But with using a high-throughput phenotyping approach, we know that the challenge with generation of complex data sets, and this requires a robust pipeline analysis to help us to get meaningful insights. And already recent studies have been working on this in finding or in combining also integrative phenotyping together with machine learning to help us to unlock these insights that we generate from the big data sets. As a biologist, the challenge is that how we can handle and interpret the large data sets. So we aim to apply the machine learning approach to high-dimensional phenotypic data to uncover these patterns, especially when we're looking at stressed plants and we want to understand better their adaptive responses. Today, as I mentioned, I will be giving a case study that was done in frame of capitalized project. This project aimed to exploit natural variation in photosynthetic traits to identify and develop new genetic resources, and also finding new tools to investigate the data. And today I will be focusing on, as I mentioned, this work was done in collaboration together with Professor Zoran Nikoluski from Potsdam University and his team, and also Professor Eyal Friedman and his team. And we had now the data is, and also the study is published, available now. But let me take you through what we have done. So briefly, the experimental design, we have done it on our modular phenotyping platform. So in this study, we have 6 barley genotypes were selected. So in total, we phenotyped 267 plants. And we are talking for phenotyping duration around 10 weeks. So almost the whole life cycle of the plant. So starting from tillering stage up to the senescence. So we classify them into early and late stage. Late stage will be mostly for the reproductive phase. And for treatment, we have control and drought. Drought stress, we already induced it from the beginning. So from the tillering stage, we already have the plants under drought stress, and we maintain this level. So we are talking about between 25 to 20% soil relative water content. And we have the phenotyping approach. We phenotyped weekly, and in each week we have 3 times RGB image, thermal IR image, chlorophyll fluorescence, and once a week hyperspectral image. And then we had the opportunity also to look at the yield data and investigate further grain-related traits. And the key questions that we would like to answer is which of the lines are most robust to the induced drought stress? Which are the key traits and temporal traits of interest that we would like to pinpoint and find them? And what we can do with the predictive model, especially we have a data set that have the phenotypic traits and also harvest traits. So the potential is huge. So with that, we together with Professor Zoran developed a kind of pipeline to hopefully it's going to be inspireful for you. So we have talking about 148 traits that was measured across 70 time points. So for that, we have two pathways, either to look at the data as a daily, so non-aggregated data, or to look at the data like a weekly or aggregated data, so taking the mean, max, and average, and then look at the data in this kind of 10 weeks. And then we tried both approach to see how the traits will be consistent over time, those informative traits, and also to see the trend. But today I will be focusing on the output from the daily time point. And now we look at the pipeline. So we have, after, of course, pre-processing data, checking for normality, etc. So we had the phase where we interested in the key feature selection. And here we are. I'm going to show you some results with k-mean clustering, tolerance ranking. And also we dived deeper and looked into data mining. And we were interested in the classification model to look at the effect of the drought stress. Also for the prediction of the harvest model, we looked also through the whole time point and also early time point. And also the script used for this pipeline is also available on GitHub. So let's get started and see the first, the overview, how was this treatment applied, how the genotype performed. So from the PCA plot, we can see clear discrimination between the control, so the blue circles, and the drought, the triangles. And also we can see one of the lines that was cluster separated. This was from a different population, so kind of reflecting a different mechanism under, regardless, of course, the stress, but it have different mechanism. Second, we wanted to do tolerance ranking to know which genotype are more tolerant. So for this, we used Prima Nova. It's to see how significant the drought from control. And we can see nicely, here we look at temporal traits, so traits from phenotyping and also harvest traits. And we can see there was significance between the stress. Second, we want also to evaluate the effect of these stress, how strong it is. So we use the effect size. And we can see also that the lower the effect size, that means the more tolerant the plants. And we can see that those that were showing tolerance among the temporal trait was also showing a tolerance at the harvest trait. So kind of combining, or the observation observed was reflected also on the yield. Second, we applied the random forest because we are interested to answer the question, which traits of interest can help us to discriminate between treatment. So in this case study, we can see that among the variable importance here, from the thermal imaging sensor, delta temperature at the early phase was showing as important trait in this early phase. And that clearly explains here in this figure that after we induce the stress, so from the second week onwards, we can see the clear significance between the control and drought. Because here, the higher the delta temperature, that means the stomata are closing. So the plant's trying to avoid the water loss, but of course that result in a bit higher canopy temperature. And when we look also at the later stage, so we can see also traits from RGB, for instance, and also a trait from chlorophyll fluorescence, like the quantum yield, was also showing importance at this at the late phase, because the stress intensity increased, so also reflect the stress. And now we move further with the temporal phenotypic prediction, because we are interested to know what we can do, or how our harvest data can, if it's informative or can be used as for prediction for these temporal phenotypic traits. So for this, we have our data set. So we train the model, our model. So we have either, you can look at the data set as control only and drought stress only, or pooled data set where we combine basically everything in the data set and we train the model and see the accuracy of the model, how much it will, the accuracy will change if I have data set that has only control or drought or pooled data set. And results show that pooled data set was showing high accuracy. And here we can see that among the harvest traits, we can see that the biomass dry weight was showing high accuracy. And this is kind of the most predictive trait based on the values we can see, followed by, here we have the tolerance spike weight and also a spike number. Where we can see here, these are kind of the strongest one. And just to double check also and see the performance. So we have also here, we looked at the biomass dry weight. If we have the model like pooled data set or drought or control, so we can see that pooled data set have higher accuracy followed by data or the model trained from the drought. And this is because, you know, under drought stress, it's better, you can see the amplification of the response and better discrimination. So that maybe explains why drought was showing higher accuracy than control. So we moved further and we take the harvest trait, like the biomass dry weight, and then we started to correlate it with the phenomic traits. And for that, we applied the lasso regression model, and we looked into the lasso coefficient, and we can see that the RGB, either like RGB top view or side view, at the late phase were highly correlated with the biomass, which of course making sense. But what also was interesting to see correlation from other imaging sensor. As you can see, for instance, there are some traits from the chlorophyll fluorescence and also other traits like the delta temperature was also showing kind of correlation. So we thought about, okay, what is the potential if we only looked at the early phase, how the model performance will be if I just looked until the stem elongation phase, or before that basically plant go into the late reproductive or reproductive stage. So here we can see I have the all data, like whole time points and early time points among those 4 top harvest-related traits that were observed. And we can see that even though the accuracy was slightly significant, but it's still like above 0.8, which is still fairly reliable. And we repeated again and looked into the impact or the correlation of the biomass dry weight with the other temporal related traits. And we can see again, it's confirming. We had the RGB, of course, it's correlating together with some traits as we see from chlorophyll fluorescence and thermal imaging. But what was interesting here that we can see traits, complementary traits from the spectral reflectance, from the hyperspectral imaging was also showing kind of correlation together with the biomass and the traits from the veneer image or the sphere image. So to sum up here, we can see with this pipeline, there's possibility that we can, by integrating this data from multiple imaging sensor, that we can find those robots genotypes, or genotype, of course, depending on the stress that induced, but in this case, the drought stress. And also we can pinpoint those phenotypic traits that are interested. And also, this study has been also addressed from the molecular point of view. So if you are also interested to look at the story from this perspective, there is a recent publication is being released from E.L. Friedman's lab. So also to provide more insights about this phenotyping approach, including some GWAS and genomic related information. And with that, I'd like to thank PSI team and also the consortium from Capitalize project. And yeah, I'll be happy to get your questions. Thank you.
So, Lamis, thank you very much for this very nice and clear and concise presentation. It's open for questions. Justin?
Yeah, Lamis, thanks. I have one quick question. You did not go into detail about how the genotypes were selected and what the material actually is. You just said one genotype was from another collection. So is that selection based on field data, contrasting for drought, or what's the background?
We have a champ population and the hip population, so they are more like we are looking at the effect of their phenology in flowering time, and there were some like tagged genes that we are trying to see if there's impact in flowering time. That was inside those genetic background of the lines.
Okay, thanks.
Okay. Rob?
So coming back to that, that separate population, was the predictive power that's used in that particular genotype or as strong in that particular genotype than the other genotypes clustered?
Yeah, for the different performance, we could see it for those that are having from different population. So this hip population that clusters separately than the others.
Yeah.
So it could be related with some mechanism.
But the question I'm asking is when did you find that the measurements that you were making that was as useful as predicting yield in that distant population, or in that genotype that was distant from the main cluster? Or was it less?
Yeah, because
the correlation is less strong in that.
I cannot recall now was in terms of genetic, in terms of grain yield, how was the performance? But yeah, we looked into this. But yeah, maybe I didn't show it here. Yeah, because the aim is that they maintain the yield or the high yield loss. Yeah, but I think overall yield we saw yield reduction under drought stress. But maybe the effect of the percentage of reduction varies between the genotypes. Yeah.
Marco, you have a question. You raised your hand, so please go ahead and ask your question.
Okay, very fast. I see that you used several tools to assess the response. So if you can rank or give a quantification of relevance among considering RGB, obviously, but I would like to assess the relevance versus infrared and the chlorophyll, which probably missed the way to capture. How did you capture the chlorophyll fluorescence or
Yeah, for the temporal trait, we actually considered all the traits from the multiple imaging sensors. So all of the information was pumped into the pipeline. So they are all included, but then we could see that most data from RGB, in this case study, was first coming as important, followed by other traits from the chlorophyll imaging sensor or the thermal imaging, for instance.
So basically, the RGB is the core, then you can add specific information from the infrared, but at the end, you cannot miss the RGB.
Yeah, because also RGB was showing importance at the late developmental stage, but the thermal imaging, for example, was showing importance at the early phase, which was reflecting the response to the drought stress. So which was interesting for us also to see physiologically, on a physiological level, how the plants are responding.
Okay, thank you.
You're welcome.
Right, then, thank you very much again, Lamis.